Hey, what's up YouTube? It's me, George, with No Copay Education, and in this video I was wanting to go over a biology question of the day regarding protein structure. The question reads as following: which of the following is most likely to be preserved when a protein is denatured? A. Primary structures, B. Secondary structure, C. Tertiary structure, and D. Quaternary structure. I'd actually recommend that you pause the video and try to answer this question yourself before playing the rest of it and hearing the rest of the explanations. Here's a very good picture, courtesy of Khan Academy, explaining the 4 layers of protein structure. I think it does a really good job with the exception of the quaternary structure. I think there's an easier way to understand it. But let's just dive into it. Primary protein structure, that refers to the sequence of a chain of amino acids. And this refers to the covalent bonds. So, for instance, glycine, alanine, valine, tryptophan. So these amino acids all have different shapes because of their different R groups, and they'll have different interactions with the rest of the amino acids in the protein. But that's not what you're looking at whenever you're referring to protein structure. Protein structure is referring to the order of amino acids. So glycine, alanine, valine, tryptophan. Let's say glycine, alanine, tryptophan. So these two different strands of amino acids are very similar because alanine and valine are very similar. So these two proteins are going to look pretty similar to each other in a three-dimensional configuration because alanine and valine just differ by the length of their R group carbon chain, but there's no difference in the physicochemical properties, really. No significant difference between the physicochemical properties of alanine and valine. But even though these two structures are going to look kind of similar in a three-dimensional conformation, these two have different primary protein structures because the sequence of amino acids are different. So primary protein structure just refers to the sequence of amino acids connected to one another. And it also refers to covalent bonds. So covalent bonds are the strongest bonds, some of the strongest bonds. They're stronger than any intermolecular forces like van der Waals interactions or ion dipole or hydrogen bonds. So now that we understand what primary protein structure is, let's move to secondary protein structure. Secondary protein structure refers to the alpha helices and beta pleated sheets, and this is really parts of the amino acids that form intermolecular forces that have intermolecular interactions with one another and sort of form these different conformations. But generally speaking, if you're talking about alpha helices or beta sheets, those are the most common forms of secondary structure that you're going to run into. Now that we understand what secondary protein structure is, tertiary structure is kind of very similar, except for instead of referring to the intermolecular forces of the backbone, it's talking about the R group. So the R group, that's going to be the side chain, and the different side chains are what make the conformation and the physicochemical properties of each amino acid different. So for instance, if you have a aspartic acid amino acid, then that is going to form hydrogen bonds with other amino acids that have nitrogen or sulfate or sulfur or oxygen. Now that we understand what tertiary protein structure is, quaternary protein structure is, I don't really like this Khan Academy explanation of a protein consisting of more than one amino acid chain. I think it's more easy to remember this by thinking about it in terms of tertiary protein structure subunits. So you can't have tertiary structure without having secondary or primary structure. But let's say you have a subunit 1 that has primary, secondary, and tertiary structure. And then you have subunit 2 that also has tertiary, secondary, and primary structure. Well, subunit 1 and subunit 2, they'll have interactions with one another. And these interactions, that is going to be the quaternary structure that's referred to. So now that we understand the 4 layers of protein structure, let's go into what denaturation means. So denaturation means destroying the weaker bonds. And this is done through several methods, including increasing the temperature incredibly high. And denaturation is just destroying these interactions between these amino acids that really contribute to the protein structure. And covalent bonds are much stronger than any intermolecular forces. Now that we understand the prerequisite science knowledge, let's go back to the question. Which of the following is most likely to be preserved whenever a protein is denatured? Well, secondary, tertiary, and quaternary structure are all referring to intermolecular forces, whereas primary structure is referring to covalent bonds. Therefore, A is the best answer. Thanks again for watching this video. If you have any questions, comments, or concerns, feel free to email us at nocopaytutorials@gmail.com. Thank you.